Yeah, I wanted to give you the color copies because for this unit, especially, some of the pictures are helpful if they are in color. So that's why you have those. Um, Yesterday, we had just gotten through 16.1, kind of an introduction to how we know um, that DNA is paired up with adenine and thymine pairing up and cytosine and guanine pairing up. And then once we found that out, then one of the next questions became, okay, so they're complementary, A with T and C with G. How is it that when we go to make a copy of our cells, we have to make a copy of the DNA first? So how is it we make a copy of that DNA? What's the process by which you somehow make make two strands of DNA from the one strand that you had when you go into synthesis of the cell cycle? How do you do that? And so one theory was that you start with a parent strand, and hopefully you can notice in there that the colors are slightly different. It would have been nicer if they used a little bit more obvious color, but we've got a blue color there. And then those two strands, those complementary sides, those nucleotide bases, the hydrogen bonds will be split apart, and you will have two strands. So the A with the T separate from each other and C with G. And then each one of those new strands will serve as a template for laying down a new strand. And if you notice the color of the new strand, they've made it a lighter blue color to show you those are the new nucleotides that are being added onto that strand. Okay, so each side of the DNA molecule serves as a template for laying down new nucleotides on the other side, the other strand. So there's, for every time you go through synthesis, then you have one side of that DNA molecule that's going to be old and one side that would be new. Okay. That's what Watson and Crick proposed. They called it semi-conservative because of the fact that each old strand served as a template. So it stayed intact. One side stayed intact, and one side was new. So semi, because one side is conserved and the other side is brand new. There were competing models that said that there were different ways that it happened. One was conservative. The conservative model said that that somehow, and it was never really quite sure how that happened, that those two parent strands stayed together and somehow created another two strands that were completely new. So you'd have a new DNA molecule that was completely new nucleotides. They would not have one side that would be old and one side that would be new. And then you had this really wacky kind of idea called the dispersive model. And the dispersive model said that somehow they were, each strand of the DNA molecule was a combination of old and a mix of old and new nucleotides. And nobody had a really good feel for how either of those models would work. But there was a guy, and by guy I mean guys, there's two experimenters by the name of Messelson and Stahl. Again, names are not important, but knowing the details from these experiments is important. So you don't have to remember Chargaff's rules, but you have to remember that whenever he looked at whatever species he looked at, the amount of adenines always matched the amount of thymines, and the amount of cytosines always matched the amount of guanines. That's Chargaff's rules. They pair up. Same thing for Messelson and Stahl. They did this kind of cool experiment where they started off with a molecule of DNA. And then they exposed that DNA to nucleotides that were marked, were labeled. They were radioactively labeled. And in that radioactivity, you can really see it in this picture. Okay, that's why I'm confused. In that radioactivity, the light color is kind of a light blue. Again, in the picture, again, it's kind of hard to see. Um, there's a light blue strand and there's a dark blue strand. So to begin with, all of the strands are dark blue because they're all old, right? Then they get split apart, and when they get split apart, what you would expect to see is one of the strands would have that old dark blue DNA, and one would have the radioactive lighter blue DNA if it was semi-conservative. And that's what they found after the first replication. One of those strands is a dark blue, and one of those strands is a light blue. Okay. That was the first time they did it. Then they allowed that DNA to replicate again, to make a copy again. And now each one of those DNA molecules acts as a template. But one of those strands is not dark blue, it's light blue, right? It's radioactively labeled, and it doesn't actually look light blue, but we're saying that it's light blue. So if it's light blue, then when it goes to replicate, the new strand and the old strand are both going to be radioactive, and they'll have that light blue color. And that's what they found, is that if a radioactive strand served as a template, then the other strand was also radioactive. And so you found that both strands had the radioactivity, and both were from the same source. And that supports the idea of semi-conservative. Dispersive model said that they somehow broke apart and came back together again in kind of a random way. That was not supported. Conservative model said that the parent stayed together, and then they somehow made a completely new copy that was all new nucleotides, and that also wasn't supported by any of the data. The only one that was the semi-conservative one here, this guy. Right? Questions on that? So these individuals are the ones that you need to know the experiments. We know what was going on. We've got Shargoff with his base pairing. We've got Wilkins and Franklin showing the X-ray diffraction, showing that it was a double helix and having a specific width to that turn. Watson and Crick putting it all together, showing that A's paired with T's and C's paired with G's, and it was in that double helical form. And then Messelson and Stahl showing that it was semi-conservative replication rather than the other forms of replication. All right. Next thing we need to get into is actually looking at how DNA does replicate itself. Now, we talked about it being semi-conservative, but there's a lot of parts to the process that we want to talk about. Copying of DNA is remarkable in its speed and its accuracy. There's more than a dozen enzymes and other proteins that all take part in the process of DNA replication, and it happens very quickly. It's going to start, replication of the DNA molecule will start at an area or areas called origins of replication. Sometimes they're called ORIs. 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 Origins of replication. And at these sites, the DNA will, the two strands will separate from each other and form a little bubble. And it's in that bubble area, that replication or that origin site, where there's actively new DNA being added on to the old template DNA. If you are talking about a bacterium, one little tiny bacteria, then you're just going to have one replication origin. That's it, one ORI. If you are talking about a eukaryote, because eukaryotes have so many DNA molecules that they have to go through, they may have hundreds or even thousands of replication origins. So ORIs, so you're going to have a lot of them because you want it to get done quickly. because the longer that DNA is open, the more chance there is for errors to happen, for damage to happen to the DNA. So you want it to happen very quickly. Replication is going to occur going in both directions of the DNA molecule. So it'll go left. Wait, that's not left. It'll go left, and then it'll go right. Okay, so it's gonna work in both directions, and that also helps to speed up the process. This next picture is of, on the left-hand side, we have a picture of a bacteria, it's E. coli, where you've just got one little replication bubble. There's one little replication bubble there. Whereas in B, we have multiple bubbles, multiple replication bubbles happening simultaneously. And this picture, not going to worry about that right now. Okay, so at the end of each replication bubble, there's going to be an area called a replication fork. It's a Y-shaped area, and that is where the DNA is going to actively be added on to the existing template strand. There's several different kinds of proteins. Some of them are enzymes, some of them are not. They're all involved in the process. One of those is helicases. Helicases' jobs are to untwist the DNA right at that replication fork area. They're responsible for breaking the hydrogen bonds that are holding the A's and T's and C's and G's together. So they're gonna open that up.
Wait, why would they want to split them?
They want to split them up because each of the strands is now going to serve as a template for adding on new DNA nucleotides. Single strand binding proteins are also, these are not enzymes, they're just proteins. They're kind of globular, they're balls basically, that hold the DNA apart. Because if you have a piece of yarn and you try to separate the strands and you let go, what happens? Doing, they want to go back together, right? Same thing with the DNA molecule. There's these two strands and they're twisted around each other, and they want to be twisted around each other. So if you don't somehow keep them open, they will try and twist back together again. So those binding proteins are going to hold them open, so that once the helicase breaks those bonds, it'll hold them open so they will not come apart, or they will not come back together, I should say. Then you also have these things called gyrases or topoisomerases. And these are to kind of relieve the pressure because you're opening up the DNA molecule in a way that it doesn't really want to be opened. And so there's a tendency for it to kind of twist, even though you've got those single-stranded binding proteins on there. And if you don't hold it really rigid and keep it from twisting, it could break apart. And if it breaks apart, then your molecule is messed up, and then you can't perform synthesis, and it's going to have to go into repair before it can go through the whole mitosis thing, or maybe not. Maybe the cell will just be destroyed if it's messed up. So we don't want that to happen.
Wait, so it keeps the DNA strands straight?
Yeah.
Okay. And then the binding proteins just make it so that they stay
straight. They don't snap back together. But even if they're not snapped back together, they tend to rotate back and forth, and that can cause them to break.
How does it keep it straight? Does it just give it more structure? Or
I think it gives it more structure. Okay. I think they physically bind to the DNA to help that.
And then it just how does it unbind?
Yeah, single strand.
I don't know for sure, but my guess is that there's other enzymes that break those enzymes apart. Yeah. This is a table showing you all these things. I know it's written really small. It is in your textbook. This is table 16.1. And we're going to go through each one of these. So these are just kind of here's what's going on. Here's the name. There are a series of enzymes called polymerases, and there's DNA polymerases and there's RNA polymerases. But in addition to just being RNA and DNA polymerases, there's different numbers and they do different things. So there's DNA polymerase 1, 2, and 3. For this class, you do not have to distinguish between 1, 2, and 3. So whenever you see that in the book, it's like DNA 1 does this, DNA 2 does this. You can just refer to it as polymerase. You do not have to refer to it as 1, 2, or 3, Paul, because that would be more than we need. Okay. Here is an example of a DNA, double-stranded DNA molecule that has been broken apart. You've got here in green, not what I wanted to do. You've got this, hold on, I wanted to get that. Yeah, there we go. In green, you have this little bubble that is going along and it is breaking apart those hydrogen bonds. It's very common to say that helicase is unzipping the genes, it's unzipping the DNA, because you're unzipping those A's and T's and C's and G's. You also have these little proteins that are shown here, and they're also shown here, that are holding it apart so that now when it's untwisted, it's keeping it from twisting back together again. And then farther back, you have this gyrase or topoisomerase. You know what the gyrate is? It's kind of like to gyrate, right? To twist back and forth. You don't want it to twist back and forth because if it twists too much, it might snap because it's being held in place with those single-stranded binding proteins. So farther up, or I should say farther upstream, you've got that topoisomerase there, and that holds that DNA in place so that it doesn't want to twist and snap. Notice we also have this thing that is called primase in pink. And primase is another enzyme that is kind of a funky enzyme that I think is the bane of many introductory biology students because it doesn't make a lot of sense, and I don't really have a reason for why. Well, I do have a reason why, but why it exists. But let's just I'm alluding to something. Let's talk about it in this page first, and maybe we can come back to that picture. So I said there's these DNA polymerases. DNA polymerases are basically responsible for the bulk of the making of the new, putting on the new nucleotides for the new strand of DNA, mainly. But DNA polymerase has this funky quirk about it. And that quirk is that it cannot start unless it has a primer added first. And that primer has to be added on. It's got to be jump started. It actually has to attach to something, and the thing that it's going to attach to is going to be that DNA polymerase. The primase. The DNA polymerase will attach to the primer or the primase. So the initial nucleotide strand is a short series, and here's where it gets. I said it's a primer, right? Well, it's a primer not made of DNA. It's made of RNA, which is a weird part. So rather than using DNA nucleotides to start, you have to attach RNA nucleotides. So remember, we're not going to use deoxyribose. We're going to use ribose. And instead of using thymine, we're going to use uracil. Okay. That is accomplished. Adding on that little short sequence of nucleotides is done by an enzyme that is called primase. Sometimes it's called RNA polymerase. Let me put that down here.
Someone sell puffs.
Well, a lot of it is what's the word I want? Not intuition. Well, no, a lot of it is they do something. Like, for example, they make the RNA polymerase no longer functional. What happens? Inference. That's what I want. An inference. So, if now all of a sudden DNA polymerase can't work, then we know that RNA polymerase had to have something. And if RNA polymerase didn't work and there's no RNA primer, well, we know the RNA primer was laid down by the RNA polymerase, and then now the DNA. So there's a lot of inference that's going on.
It's not like they're like seeing it.
Yeah,
like
a lot of times they're not seeing it.
And then they see the result. And
Yeah.
Figure out
Yeah. Lots and lots of inference in science at this level because it's just too small to see. Okay.
Repertoire is like if a vernacular is the vernacular for language.
All right, so this is just showing you. I don't know exactly why this is in here, but it is that difference between DNA and RNA. Remember, RNA has the ribose. DNA has deoxyribose, obviously. And we also have the uracil replacing the thymine in DNA. So once that primer has been put in place, now those enzymes called DNA polymerases, now they can take over. And they can start adding on. They will physically attach to that primer, kind of use it as a crutch. And now they will attach the DNA nucleotides along that old template strand. Most DNA polymerases require a primer, and they also have to have a template strand, right? If there's no template strand, they can't add anything on. This just like I don't even know what to make of that. Elongation, that just means how fast they put them down. Here they're saying that about 500 nucleotides per second in bacteria. And in eukaryotes, it's in human cells, it's about 50. So it's slower. Wow. It's only 50 per second. But that's really, really fast. So you're adding on things in a very quick manner that makes it less likely that replication bubbles open for too long and something happens, like the DNA sequence gets messed up. Okay, so each nucleotide that is added to the growing DNA strand is called a nucleoside triphosphate. Yeah, we don't need to really worry about that. Just trying. No, we just don't. Yeah, no. Let's. Yeah, no. Don't need to know that.
Sir, I like that.
You're welcome.
Thank you.
All right, so remember we have an old strand that's dark blue and we have a light strand that's light blue. And here we can see that when we have the DNA polymerase, we're adding on a new incoming nucleotide here. And that's really all you need to know from there. Okay, now here's one of the key annoying parts in addition to the whole primer thing. Remember how I talked about this whole anti-parallel thing? Well, you remember how enzymes have a certain shape and they're very specific about that shape? Well, think of a puzzle piece. And a puzzle piece fits in a certain way, right? But if that puzzle is now flipped upside down, the puzzle piece isn't going to work. What do you have to do to the puzzle piece? You have to flip it. So you have to flip the enzymes as well. What? What? I'm so confused. Same. I'm trying to figure out if I can do this on this slide or if I need to. No, I haven't done a puzzle upside down. That sounds terrible.
I mean, you can do it. You just have to
Let me put in a slide here.
That's like doing the
All right.
Someone did that. Yeah, I mean, you can do it. You just have to basically try everything.
That's what I mean. I'm called like that.
Okay, I have a couple of strands of DNA here. Pretty poorly drawn. I can't even draw them in there. They're so tight together.
Definitely not Jim.
Okay, you get the idea, I hope. Okay, enzymes, DNA polymerase, as well as primase, will only work by adding on nucleotides onto the template strand from the 5 to 3' direction. So it has to start here and adds on nucleotides here. That's the enzyme. That's my DNA. Well, let me put my primer in there too.
Well, guess like you're wrong. I'm black.
Well, but when you look at the pictures, it looks different than that because it's adding it onto the 3'.
Wait, I'm actually confused too.
Because it says it adds on.
Sorry, I did that wrong. I did that wrong.
Just like I was wrong.
You weren't wrong. I was wrong. Sorry. So,
It goes in the 5' direction. So it has to
right. Thank you for catching that. So it's going to add them on 5 to 3. So here is 5 to 3. And this one is 3 to 5. So when I'm adding this one, I'm working towards the fork.
Bro, what?
So one is being.
It's always 3 to 5.
It's always 3 to 5. So one is working towards the fork, and one is working away from the fork. Because they're going in opposite directions. And they have to go in the same direction as the strand is going.
Wait. But then how does the one that's going like on the bottom, how does it start from the end then?
Good question. So this is only part of the story.
That'll go over.
Yeah. So Tyler's question is a good one. When this DNA polymerase gets to the end here, it's like, what do I do now? I'm out of DNA. What does that polymerase have to do? Yeah, it's going to jump backwards. And it's going to start here. And it's going to add more on.
I feel like that's
Wait,
like
is it making the DNA? Is that what's happening? No way. Is it making it?
Yeah, it's making DNA.
Oh, lock in, bro. What are you like?
Okay, that just clears everything.
Dude, what did you think you were looking at, bro?
So give me a second here to add this on again.
No, I mean that makes sense actually. No, I still don't get it. Why is it still like it's like burnt into the screen? I can see it.
Oh, yeah, it is.
It's actually so
Well,
weird.
interesting.
Not that weird. It's not that funny.
So we still got these guys back here.
Now you can just tell us what to do.
Oh.
He's so tough.
I'm literally about to explode. What is it?
You're going to explode? Oh, I didn't want to do that. I'm going to try and do this.
So foul.
No, that isn't what I wanted. Why rectangle? Why? There we go.
Yeah, I'm not gonna lie, bro. You gotta go. That's a wrap. Another miss, bro. Another miss, bro.
But even the bitches are so
I just don't think they were laughing. Like,
No, but it was so funny.
the freestyle.
So annoying.
No, usually you would actually see. Why would you do it?
And I asked
What did I say?
All right, so in this strand, I'm going to start off and I'm going to make
It's just. Yeah, I can say.
this is to look at. Wait, what happened to my other strand? Did it just disappear? Dang it.
Yeah, I feel like.
No, because then I undo all my writing too.
I feel like the technical issues just like mean that it's time to take a break from notes.
I think it's time to
Study.
All right, look at this up here. All right, look at the arrows. One's going towards the replication fork, so one's going down here towards the replication fork, and one's going away. So this one that's going towards the replication fork, as this piece continues to unzip with the helicase, it just continues to add on DNA nucleotides.
The one on the top just keeps going.
Just keeps going.
And then on the one on the bottom, it just keeps jumping back.
Right. So the problem with this one is he's done. He's out of DNA that he can work with. So then he's got to jump back here.
Wait, Doxy, what happens when he like? Does it go over the DNA that's already created, or when it stops?
It stops.
Okay,
So it'll stop there.
so it doesn't like go to the end.
Right. So then it's like, okay, now what do I do? So now it's got to jump back again, and it's got to start over again.
Isn't that like super?
It keeps pulling apart. It's like it's slowly being pulled apart. And as it gets pulled apart, it's like, oh crap, I got to go back here. And it's like, then it pulls it more apart. It's like, oh, I got to go even farther back. It pulls it more apart, and it's got to go even farther back.
It's like the most.
Yeah. Yeah, people don't really understand why this happens. What does it do? Well, it's putting those blue pieces that I'm showing you, those are new DNA nucleotides that I'm adding on. Yeah. No, you're not the only one, Nate. Other people have said that too. Like, what's going on? All right, so this is the better picture that I didn't want to show you just because it's so small. But let me see if I can. Let's see, I want to zoom out, right?
Wait, Doxy,
Yeah.
on the slide, the intratrail elongation slide, it says, therefore, a new strand of DNA can elongate only the 5 to 3 times.
Yeah, it's adding, that's the other problem. It's adding on new nucleotides, starting from 5 and going towards 3. But it's matching up with the other strand that's going from 3 to 5.
So, like, the template is 3 to 5?
Mm-hmm. Yep, and the ones that are being added on are 5 to 3. Right. Template strand is 3 to 5. The new DNA nucleotides that are being added on are 5 to 3. Because it has to match, right? DNA polymerase can only work in the direction of 5 to 3. So this picture is just showing you that top strand. And it's working towards and it's showing you a couple of pictures here. The first one is, we'll call this time 1. And then this one down here is time 2. Maybe one's 1 minute. Well, actually, not even 1 minute because we know how fast it goes. But maybe 1 second is time 1 and 2 seconds is time 2. Okay? So it's moving down towards the replication fork on that top strand. We call that top strand anybody know? Leading. We call that the leading strand because it doesn't have to wait. It doesn't have to pause while the helicase unzips anything. It just continues straight forward. It doesn't have to jump back. It does its job faster than the other strand does. The other strand is going to be called the lagging strand.
Does that like affect anything? Or is it just like that they're not on the same
It
side?
does. It makes more work for the lagging strand. So what we're not going to get to today is the work that the lagging strand has to do to finish up. It has to do a couple other steps to deal with all the things that it has to do.
I just like that's so like I feel like the body's kind of dumb for that.
Yeah,
How is that like
I don't know if anyone really knows. I mean, one of the theories is that we actually didn't start out as living creatures with double-stranded DNA. It was actually RNA. A lot of people think it was RNA, and RNA single-stranded. So if you're using the same enzymes, but now you've got two directions that you got to work.
That's like how big? What? Like we were humans that used RNA. Like
Well, no, we wouldn't have been humans. We would have been like single-celled organisms back in the day. But we would be using RNA, and RNA only has one strand, so there's no direction issue. And I'm really guessing on that, but I think it's a decent guess. All right, I'm going to stop there. That's the hardest. Well, this is the hardest bit we're getting to. It takes some thought into getting this. If I have a great assignment for you, if it's a snow day on Friday. Well, everyone's like speculating. I have a great one for Tuesday as well.